All right, good morning, everybody. Today we are going to be learning about translation. So first thing that I wanted to remind you about is that you need to turn in your transcription homework from yesterday. So if you did not do that yesterday, I would start class by getting that done and turned in. When you are done with that, it says translation checkpoint, but you're supposed to take the transcription checkpoint in Schoology. So that is a little checkpoint over what we learned about yesterday. Make sure that I'm still sharing my screen. Okay, there we go. So when you're done with the checkpoint, then you are good to come back to this video and fill out your guided notes. So today what we are focusing on is translation. Yesterday we talked about transcription, which is the step of turning DNA into mRNA. And mRNA's job is to carry the code of that one gene to the ribosome so that it has the instructions. Translation, which we're talking about today, is how the ingredients get brought to the ribosome so that we can actually put that protein together. Transcription is the step in protein synthesis where blank makes blank. What do you think? It's where DNA makes mRNA. I kind of feel like Dora, where I leave spaces and then nobody responds. Anyway, I'm going to keep going. This process takes place in the nucleus. It has to take place in the nucleus because it involves DNA, and DNA is trapped there. After transcription, the cell has a really long strand of mRNA that serves as a messenger between the nucleus, where the DNA is, and the ribosome, where the protein gets made. True or false, before translation, the cell edits the mRNA sequence. That is true. Blank are the sequences that were cut out. Those are called introns. I know it sounds opposite of what it should be, but introns get cut out, and exons are the sequences that get left in. When the strand has been edited, the cell splits the bases into groups of 3. Each group of 3 bases is called a codon. And each codon codes for 1 amino acid, which is why when you put those 3 letters in the chart, you end up with something on the outside of the chart. That something you end up with on the outside of the chart is the amino acid. When these are put together, they make a specific protein. Remember that transcription had 3 parts: initiation, which is where the promoter sequence on the DNA says this is where the gene begins; elongation, which is where RNA polymerase makes a copy of the RNA; and then termination, which is where the DNA has a terminator sequence that says the gene is done, go to the ribosome. Exons get expressed, introns get cut out, and the mRNA gets read in groups of 3 called codons. We did some practice with this yesterday. AUG is an exon, so we know that we have to turn it into an amino acid. You start with A, then you move to U, then you go to G, and you see that it codes for methionine. Methionine is special because it is our start codon. Every protein starts with the start codon. Then we notice that AUC is an intron, so it will not turn into an amino acid. UCG is an exon, and we go to our chart. UCG codes for serine. And then our last exon is UAA, which codes for stop. That is a stop codon, which gives us the information that the protein is now done. So these are our amino acids that would make our protein. Remember, each amino acid has a different R group. The order of the amino acids determines the proteins. So if we were to switch any of these amino acids, then you would have made a completely different protein. So now we're good to talk about step 2. Step 2 is translation. Translation is where we're actually going to put together the ingredients to make the protein. At the end of transcription, your cell has made an edited strand of mRNA. Translation is the step that takes the mRNA code and turns it into a protein. Now, this is kind of a silly picture, but it is a little bit what the ribosome actually looks like. The ribosome has two subunits, an upper subunit and a lower subunit, so it's kind of going to look like this. The mRNA is going to go in, and the strand of amino acids, which is the protein, will come out. If translation makes proteins, where do you think it happens? Think about which organelle's job it is to make proteins. If you thought ribosome, you are correct. Translation occurs in the ribosome. Again, you can see how the mRNA enters the ribosome, and the protein made of amino acids is what will come out. So here we see our ribosomes with the mRNA going through them. That mRNA is made by transcription, and we see the amino acid strand or the protein coming out, and that is made by translation. So transcription makes mRNA, translation makes the protein. mRNA travels to the ribosome and it waits for tRNA. Remember that tRNA is transfer RNA. Transfer RNA is who we send to pick up the ingredients, right? Just like yesterday in our analogy about making cupcakes, how I sent my husband to the store to get the ingredients, tRNA essentially does that. It gets the instructions from mRNA about what it needs to bring, and then tRNA goes to the cytoplasm and it looks for that amino acid. It grabs onto that amino acid and brings it back to the ribosome. tRNA transfers the anticodon and the appropriate amino acid to the mRNA sequence. So tRNA brings back the amino acid. Now there's something else in there called an anticodon that we're going to have to talk about in a second. But basically the anticodon is what connects to the codon on the mRNA. Step 3, amino acids are joined together with peptide bonds by the ribosome to make a protein. tRNA acts like a molecular bridge that connects the mRNA codons to the appropriate amino acid. If we look on our mRNA, our first codon is AUG. So tRNA is going to have to connect to that. So the anticodon would be UAC, because that's what would bond with and connect to the codon. So this is our tRNA molecule. Notice it has two ends. One end has the anticodon, which tells the tRNA where to go and what to connect to. Notice if the anticodon was ACC, then the codon that it connects to has to be UGG. And if you follow that, you see that the tRNA is going to the codon that's UGG. Okay? On the other end of the tRNA, there is an amino acid. So the tRNA connects the appropriate amino acid to the codon that codes for it. One end of the tRNA has the anticodon, and the other end has the matching amino acid. Now remember, mRNA is what codes for the codon. That is the code. Okay, that's what you're going to put into the chart every single time. mRNA is the instructions, all right? tRNA is just used to connect to those instructions. So you never put the tRNA anticodon in the chart. You only ever put the codon in the chart. An anticodon is the complementary codon to the original mRNA sequence. It is used to match up to the correct part of the sequence. It is basically an address that tells the tRNA where to deliver the package. Anticodons are like placeholders that allow tRNA to bind to the mRNA. Do not get this confused. The mRNA is what codes for amino acids, not tRNA. mRNA is what codes for the amino acids. tRNA has one job, and that is to transfer the amino acids that the mRNA coded for to the ribosome. mRNA is the instructions. tRNA just delivers the ingredients. The ribosome will then put those amino acids together to make a protein. Where does transcription occur? The nucleus. Very good. And that's because transcription involves DNA, and DNA is only found in the nucleus. What does transcription make? Transcription makes mRNA. What is the function of mRNA? To bring the code for the protein to the ribosome. Where does translation occur? Translation occurs in the ribosome. What does translation make? The protein. What is the function of tRNA? To deliver the ingredients, the amino acids. The function of tRNA is to deliver the amino acids. This is supposed to be a video. Let's see if I can get it going.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
There you go. Hopefully that was a good review of everything we talked about. Which best describes the process that what best describes the process that occurs when DNA codes for a protein in the cytoplasm? Is it that one strand of DNA leaves the nucleus? You should read that and immediately go, no, because DNA does not leave the nucleus. Next, mRNA is transcribed from DNA and leaves the nucleus to be translated by tRNA in the cytoplasm. Let's leave that one. C, a ribosome makes an RNA copy of DNA in the nucleus, then returns to the cytoplasm for protein synthesis. No, the ribosome does not make that copy. It is an enzyme called RNA polymerase. And then D, the tRNA copies the DNA in the nucleus. No, gotta be B. The tRNA does not code for the amino acid. The mRNA does. Make sure you don't get that confused. Anticodons are just placeholders that allow tRNA to bind to the mRNA. Now, do you think that this has happened correctly for every nucleotide that has been brought to the ribosome perfectly, without error, every single time? No, right? We make mistakes even when we are doing this and saying that A should go to U and C should go to G. Sometimes we say something wrong. Sometimes things go wrong in your cell as well. That is called a mutation. A mutation is when the DNA sequence gets changed and read as something else. The DNA sequence makes the mRNA, which provides the instructions for how to make a protein. Therefore, if there is a change in your DNA, then you will completely change, or you can completely change, the protein that is being made. Mutations can occur due to lots of different things. Some things that can cause mutations in your DNA are certain chemicals, certain pollutants, radiation, and sunlight. Sunlight can cause mutations, which can cause skin cancer, which I'm sure you guys are aware of. Radiation, which is what Rosalind Franklin worked with, can cause cancer. And chemicals can also interfere with your DNA processes. Mutations can occur during DNA replication, which is where DNA makes a copy of itself. And mutations can also occur during protein synthesis, which is where the DNA is getting turned into a protein. There are a couple different types of mutations that we are going to learn about. The first mutation is called a point mutation. A point mutation is what happens if one nucleotide gets switched out for another nucleotide. Our example here would be like if the sentence or if the sequence was ABCDEF and it changed to ABYDEF. It changes just one single nucleotide, just one point in the sequence, which is why it's called a point mutation. You can see up here that the original DNA was TAA, CTG, CAGGT. And there was a change at one point where the thymine became a cytosine. That is called a point mutation. A point mutation can be classified into a few different types. There is a point mutation called a silent mutation. There is a point mutation called a missense mutation. and there is a point mutation called a nonsense mutation. Now, it kind of seems like a point mutation wouldn't have a big effect because it's just a change in one single point of the DNA, right? Genes are made of hundreds of nucleotides, so changing one isn't going to make that big of a difference, right? But if you change a codon, then you could change the amino acid. And what happens to a protein if you change the amino acids? You make a whole new protein. So we've talked about the disorder called sickle cell anemia before, which is where instead of your red blood cells looking like an inner tube, your red blood cells instead look like a crescent moon. They're not able to carry as much oxygen. Sickle cell anemia is caused by a point mutation. The difference between normal red blood cells and sickle cells is one nucleotide that got switched out. So it can have a big effect. But it can also have no effect, which is a silent mutation, which is a little bit crazy, okay? A point mutation can be called a silent mutation if the amino acid does not change because of the mutation. Okay, so what would that look like? Let's say that the DNA sequence started as GGT and it got changed to GGG. All right, can we put these letters into the chart? No, that's because this is DNA. Okay, so to figure out if this changes the protein, we need to change the DNA to mRNA, the mRNA into our amino acids. If the amino acids are different, we know that it made a different protein. If the amino acids are the same, that is a silent mutation, and the same protein would be made. So the mRNA sequence would change from being CCA to being CCC. CCA codes for proline. CCC also codes for proline. So this mutation would not change the protein because they are the same amino acid sequences. And if the amino acids are the same, the protein would be the same, and you would never know that this mutation happened. So that is a silent mutation. In a silent mutation, if the amino acid stays the same, then it will not change the protein. Our next type of point mutation is called a missense mutation. In a missense mutation, it will cause the amino acid to change, and therefore it will make a new protein. So there would be a difference in the individual. That would be like if the DNA started as GGT and it got changed to CGT. Notice it's still only changed at one single point. So it's still a point mutation, but this is a missense point mutation. This would change the mRNA from being CCA to GCA. CCA codes for proline, GCA codes for alanine. So this would change the entire protein, so it is called a missense mutation. Our last type of point mutation is called a nonsense mutation. In a nonsense mutation, the amino acid gets changed to becoming a stop codon, and it causes the protein to stop being made early. So let's say that the DNA sequence started as ATG, and the DNA got changed to ATT. So ATG got changed to ATT. Remember, we still have to turn this into mRNA. So the mRNA would have originally coded for AUC. AUC codes for tyrosine, but it got changed into UAA. UAA codes for stop, and this will cause the protein to stop early, and therefore it's going to be very different from what it was originally supposed to be. That is a nonsense mutation. So if it doesn't change the amino acid, it is a silent mutation. If it changes the amino acid to something else, it's missense. And if it causes the protein to stop early, it's nonsense. Those are all point mutations. There's another big category of mutations, and those are called frame shift mutations. Frame shift mutations are when a nucleotide is either added or inserted or taken out or deleted from the DNA sequence. Frame shift mutations are going to change the number of nucleotides. It would either add a nucleotide or delete a nucleotide, which is why they're called insertion or deletion. So if you look, this DNA started by having this DNA started by having 6 nucleotides, and now there are 7 nucleotides. So instead of the codon being A, B, C, and the next codon being D, E, F, the first codon is A, B, B, and the second codon is C, D, E. So the frame shift mutation will affect not only the area where the mutation occurs, but everything after it as well. All of the codons after will change also. So example number 2 is it was supposed to be A, B, C, and then D, E, F, but we subtracted, we did a deletion, and now it is A, C, D, E, F. So you notice it changes the first codon, but also every codon after. So would a point mutation or a frame shift mutation be worse? A frame shift mutation has a much bigger effect. So if we did a point mutation to this sentence, the fat cat ate the rat, and we switched out this one single base here, the sentence now says the fat bat ate the rat. So it changed part of the sentence, it changed one of the words. Let's do a frame shift mutation at the bottom. The fat cat ate the rat. If we got rid of that F, this is what the sentence now says. What had a bigger effect? The frame shift mutation. And that's because the frame shift mutation affects everything after the point of the original mutation, but the point mutation only affects that one base. The frame shift mutation affects everything after. There's another type of mutation called an inversion. An inversion is where the DNA sequence actually gets wrapped around itself and it will switch a section. So instead of being A, B, C, D, E, F, it becomes A, B, C, F, E, D. So you can see it's just part of the DNA gets swapped around. Why could mutations be good? So we know that mutations change the protein, but sometimes that change in protein has no effect, sometimes it has a good effect, and sometimes it has a bad effect. So this is an example of a mutation that has happened in moths. The original moth looked like this, and then there was a mutation that caused the moth to look like that. And that is a helpful mutation because they were able to hide from predators. Mutations can actually help an organism by increasing their fitness, which is their ability to survive and reproduce, and it can also increase variability within a species. And we know that having as much variation as possible can be helpful to a species. So some mutations are negative, right, like sickle cell anemia, but some can be positive. For example, we have grown seedless limes. That was a mutation, but we find it helpful because we don't have to deal with seeds in our limes anymore. So on occasion, cells make mistakes in copying their DNA. An incorrect nitrogen base can be inserted, or a base can be skipped altogether. These mistakes are called mutations. Mutations are changes in the genetic material of the cell. Mutations can either be gene mutations, which is all of the mutations we just talked about, okay? When you change one portion of a little bit of DNA, whether it's in point mutation or a frame shift mutation or an inversion mutation, you're just dealing with one gene. That is a gene mutation. There are also chromosome mutations, which are what we talked about with the karyotypes. So if we're talking about trisomy or monosomy, those would be chromosome mutations because that's a difference in an entire chromosome. Gene mutations produce a change within a single gene. Chromosome mutations change a whole chromosome. Importance of mutations. Most gene mutations are actually neutral, meaning they don't have effect. Some mutations can cause dramatic changes in the normal cell functions and can result in a genetic disorder. Mutations that occur in your body cells only affect that one organism because they are just in your normal body cells. However, if you have a mutation in your gamete, then that can be passed on to your children. So mutations that occur in your gametes or in the production of your sex cells, then that mutation can be passed on to future offspring. And that can affect what the offspring look like within a population. If this mutation creates a favorable change, then those offspring might be more likely to survive in their environment, which can lead to evolution, which is actually our next topic, which is pretty fun. So if it occurs in a body cell, it cannot be passed on to offspring. And if it occurs in the sex cells called gametes, then it will be passed on to the children. Okay. If you guys have all your notes filled out, you've done your homework from yesterday, then the sub has a worksheet for you guys to get started on. I do not think you guys are going to finish it today, so there will probably be a little bit of time for you to work on this tomorrow. You're not going to have all of class tomorrow, so you should get a decent of the way through today. But what you're going to do is a worksheet about mutations. So this is the original DNA sequence. It's also on your worksheet. This is what your DNA started as. So if you want to leave like this screen up on your computer while you are working on this, this is what the DNA was supposed to be, okay? And you are going to have to look at some mutated sequences and tell me what type of mutation it is, all right? You're going to have to figure out if it is a missense, if it is a nonsense, if it is a silent mutation, if it's an insertion, or if it's a deletion. There's going to be one of each. Hint, only put mRNA into the codon chart. Do not put the DNA in there. There's one of each mutation, so you can check your work there. And something that I like to tell people to look for is if you're deciding whether it's a point mutation or a frame shift mutation, count how many bases there are. The DNA started with 18 bases. Okay, so if the mutated sequence also has 18 bases, then you know it's a point mutation. If the mutated sequence has 19 bases, you know that it's an insertion mutation because you added a base. If the mutated sequence of DNA has 17 bases, then you know it's a deletion mutation. Okay, so I want you guys to work on that worksheet for the rest of class. You guys can email me if you have any questions, and I would be happy to help you out. I hope you guys have a great day.